Hi guys, today we're going to cover transcription and translation. We're finally here where we're going to see how we are going to make our proteins. So from the genes, we're going to say at the molecular level how we get to these proteins. So before we get started, as always, let's do a little bit of a recap of what we covered last lecture. So we looked at DNA replication, right? So we're still talking about genetics and how important are our genes. And as you can tell, everything is connecting, right? So everything we have been looking at throughout this entire semester is really coming together. So we looked at we talked about how during interphase the cell goes through DNA replication. And so last time we were seeing at the molecular level how DNA replication works. How does it happen? And so what we know is that the process is a semi-conservative process, which means that from your original molecule of DNA, you get two new molecules of DNA with one original strand and one brand new strand. The blue one is the brand new strand. And we said, as a reminder, how is the structure of DNA? Remember, we talked about this when we were doing the macromolecules. So make sure you go back to that lecture if you don't remember the details of how the nucleotides are made. What we really focused on last time was the directionality, right? We said we have two strands of DNA. They're anti-parallel, which means one strand goes in one direction and the other one goes on the opposite direction. So as an analogy, think about the freeway, right? You have two streets and the cars goes in opposite directions. So it's kind of the same idea. You have your nucleotides that are going on opposite directions. So in this strand here, we can call it the leading strand, or the lagging strand, actually, sorry, is from 5 to 3. How we give these numbers is based on the position of your carbon in your sugar. So that means that this carbon here is in the 5th position and is bonding with the phosphate. On the other hand, this one down here is bonding with the OH group. See, OH group. So the 3rd prime, it has the OH group here, and then that H will bond with the next phosphate down here. But the carbon is bonding with the OH group, okay? So on the opposite side, it's anti-parallel, so now what it was here in 3 prime is now in 3 prime. See this carbon here, they're both in 3 prime and they're bonding with the OH, and your 5th, the 5 prime end is bonding with the phosphate group, okay? So that is how you kind of see the directionality of the molecule. And we said it's very important when we talked about DNA replication, because the enzyme that synthesizes, the enzyme that is adding those nucleotides, that is extending a new strand, that is called DNA polymerase, can only extend in one direction, which is from 5 to 3. On the new strand, right? So this is where it can extend. This is where it can add those new nucleotide at the 3 prime end position. We talked about why that is happening, so go back and check that. And so we saw how nature is so clever and found a way to be able to still extending and synthesize in the right direction, even in the anti-parallel strand, right? And we said those are the Okazaki fragments. And so instead of doing one long new strand in the opposite direction, DNA polymerase that just does multiple different Okazaki fragments that then are going to be joined together. by another enzyme that is called ligase. Okay, and then at the end we talked, of course, we talk on how we can, there are different ways to proofreading and to make sure we have mismatch repair, right? To make sure that our DNA after replication is as close as possible. It should be genetically identical to the original molecule, right? We don't want to have mutations when this process is going on. But we also acknowledge that mutation can happen, and there are different type of mutation that can happen. I'm not going to talk about this. The only thing that I want to point out is, remember that first mutation we talked about called silent mutation. So it's part of the point mutations, which means only one nucleotide changes. But in this case, it's silent, and we're going to talk more about this. For now, just remember that we said last time that with this type of mutation, even if you have a nucleotide that changes, you don't have the change in the amino acid, right? This is Val, and this is still Val, even though here you have a T instead of an A. So this is called silent mutation, which means it's not affecting your protein sequence. And that is important because we know your protein is your phenotype, right? So if your protein is affected, now you can have problems, right? Remember cystic fibrosis, for example. Those channels don't work well. Why? Because the protein level is affected, okay? Okay, so let's get started. So today, as I was saying at the beginning, we're going to finally see how we move information, so how we make our protein from our genes. We have been talking about this on and off throughout the entire class, really. But today we're really going to focus on the molecular level, on the processes. How do we do it? How do we make proteins? So this is called the central dogma of biology. And what this states is that the flow of information has one direction, and it goes from DNA to RNA to proteins. We already saw this. I already mentioned this at the beginning of the class when we talk about the unifying theme of biology. But today we're really going to dig in the specifics. I want to do a 1 second connection with some terminology that we cover during genetics. Remember that DNA, this part here, is your genotype, and the protein is your phenotype. We already said this many times, but I want to kind of remind you now, because in the flow, in this central dogma, what we're saying is the flow of information goes from genotype to phenotype, okay? So from your DNA to your protein. And how it does it is through these different steps. So those are the different steps, the different processes that needs to happen with this order to be able to get to the proteins. So let's see what are these processes. So the first one is called transcription, and transcription is at this level. So it's how we make our RNA, okay? So we're here, the blue strands are your DNA, and remember, we want, when we make a protein, we want to express specific genes. Remember, we talked about this when we talk about the macromolecules, really, and even the first class. So you have, and also when we were talking about genetics and the pedigree and so on, when we look at when we're looking at our phenotype, every phenotype, every protein is linked as being made by specific genes, which means you have a gene that is coding for the color of your eye. You have a gene that is coding for how tall you are. You have a gene that is coding for the color of your skin. You have a gene that is coding for, I don't know, the shape of your nose, the shape of your ears, and so on, okay? So it's not your entire genome. Your entire genome has millions of genes, and each gene is coding, meaning is carrying the information, is carrying the recipe to make that specific protein, okay? So when you do transcription, you are targeting a specific gene on your DNA. And what that means is that you are going to transcribe that specific gene on your RNA. So now you make RNA that is carrying that information, right? It's carrying it on its new strand. And that RNA is going to be processed if we are talking about eukaryotes. Don't worry about that. Right now, we're going to talk all about this. But anyway, that RNA is going to be the main player in translation. So that RNA that was here in red, or what is this, pink, is the same that we see right here. So this is the mRNA that was transcribed at this level, okay? So we are going to look at the details of these different processes and see how they happen. The idea that I want you to keep in mind is, let's pretend that we have our great grandma gave us a special book, you know, one of those ancient books, you know, those beautiful ones that are very fragile. The pages are like very fragile, and they have and this book is very fragile, is very important, has been passed on from generation to generation, and that is a book that contains tons of different recipes, okay, special recipes, and we love this book and we want to pass it to the next generation. So this book cannot be ruined in any way possible, imaginable. That book, in this analogy, is your DNA, okay? Your entire genome has a bazillion different recipes, it's special, it's fragile, we don't want it to get ruined, and so we keep this book in our bedroom at all times. That book never leaves our bedroom. In our case, in analogy, that would be the nucleus, right? It never leaves the bedroom. But what happens if we're hungry and we want to make a special meal? We need to carry the recipe to the kitchen, right? But we don't want to bring the book there. We don't want the book to get stained. We don't want the book to be ruined. We want that book, that DNA, to stay where it is, safe. So what we are going to do, but we do have to eat, so what we're going to do is take a piece of paper, transcribe on that piece of paper that specific recipe, transcribe it. That piece of paper now becomes your RNA, and that piece of paper, it's fine to leave the bedroom and go into the kitchen. It doesn't matter if it gets ruined after we made the recipe. And actually, we're going to throw it away. We don't even need to keep it, because once we made the recipe, we don't need our recipe there in the kitchen. And that is the same that happens to RNA. Once we made our proteins, the RNA goes out in the same way. So we transcribe that recipe on our piece of paper, and we bring this recipe to the kitchen. Now, we have the recipe, but we have to make the food, right? We have to make our protein. So to be able to do that, we have to translate our recipe in the language that is going to allow us to make the food. From words, now we need stuff, right? So we need to do that translation, and we have to make our food, okay? So that is kind of an analogy that I want you to keep in mind when we go in the details of the processes. You need to understand, as always, the big picture, okay? DNA is precious. It's been passed on generation after generation during our, we do meiosis, the gametes, and so on. We don't want to be ruined in any way possible. So when we want to express one specific gene, we just transcribe that gene and we move that RNA that now has the transcription of the gene, so we can work with that and we don't touch the DNA. The DNA stays safe and intact where it is. Okay? Okay. Let's go and see, before we go in the details, we need to do a step back because living organisms are complex and we are different. Even though we said at the beginning of the class, evolution is one of the theory of biology, right? And is definitely the unifying theme, right? We said evolution explains why we are so different, but also why we have so much in common. We will see today how many things we have in common with prokaryotes at the molecular level, which is an explanation of evolution, right? We all come from the same common ancestor, but we went through evolution, and so we have also some differences that are important. So as a reminder, and please go back if you don't remember this, eukaryotes are way more complex. Usually they're bigger. Many eukaryotes, like humans, are multicellular organisms. We have this nucleus inside the cell, and our DNA is there protected in the nucleus, right? Prokaryotes, on the other hand, are less complex. We're talking about bacteria and archaea in this case. They have usually a circular chromosome in the middle of the cell. They don't have a nucleus, but still their chromosome doesn't move around. It's not in any position of the cell. It's in a specific position. Because the structures of the cells is different, and because obviously this organism went through generation and generation of different evolution, the processes are going to look slightly different, even though the main picture is actually pretty identical, which is kind of amazing to me, right? So let's look now at the differences at the molecular level. So we're talking about how we make proteins. So let's look at this, okay? So let's start with the bacterium first. So these are prokaryotes, right? So we just said we have a circular DNA, right? DNA is not in the nucleus, but it is in a specific position of the cell. And so because of that reason, the process of transcription and translation are less complex in prokaryotes than in eukaryotic cells. Something to notice is that because there is no nucleus, you actually can have transcription. So this is the blue one is your DNA, the circular chromosome, right? This purple one is your RNA that you are transcribing. So it's the transcription of that specific gene. And then here is the ribosomes and the protein during translation, okay, that is in the cytoplasm. So because everything is in the cytoplasm, in the bacterium, you can have transcription, this one here, happening at the same time then translation would happen, okay? We're going to go in the details of that, but for now, just keep that in mind. On the other hand, in eukaryotic cell, as I was saying, we have way much more complexity, and we have something in addition, which is the nucleus. This green part here is our nucleus. So first of all, our DNA is not circular, right? We have it's linear. You have many different chromosomes as well. If you think about humans, 46. You are transcribing a specific gene on a specific chromosome at the time. and you are inside the nucleus. You are not leaving the nucleus. The DNA never leaves the nucleus. It never leaves our special room, right? That book never leaves our room. The DNA never leaves the nucleus. So what happens is that you're going to have transcription happening inside your nucleus. And right away, you can see a big difference beside the location, a big difference between eukaryotes and bacteria. The first one is, look at this. See, this is your mRNA, but look, here is all nice and purple, the same light purple. Here you have different colors, right? You have some parts that are light purple, but others that are dark. And so the reason why it looks different is because after transcription in the eukaryotic cell, your RNA needs to go through processing. This is not yet your mRNA. This is a pre-mRNA. So we're going to see in the details how we make this light purple and how we add this yellow dot here and these three green dot at the end here. We're talking more about that in a few minutes. For now, just realize that there is some level of complexity here that the bacteria don't have, okay? And then once this mRNA is ready, it's going to get out of your nucleus, right? It's going to the kitchen, and now starts translation. So definitely two processes that are very distinct. They happen in two different locations of your cell. In the bacteria, prokaryotes, these two processes can overlap because they are happening kind of in the same area. They don't have a nucleus. Okay. Let's go in the details. Let's look in the details at the molecular level how transcription happens first, okay? Because we said the flow information goes from DNA to RNA, RNA, proteins, first step, transcription. How does it happen at the molecular level? So the first thing that I want you to know is that during this process, we are making the molecule of RNA, okay? These processes resemble a little bit DNA replication. There are some similarities, but it's important that you don't confuse the two because the goal is completely different and what we're trying to achieve is completely different. So we are not addressing the entire genome. Again, here we are trying to express specific genes. So we are going to get a signal in our cell that says, hey, I need more pigments. Hey, I need DNA polymerase. Hey, I need a specific enzyme because I'm going to do cellular respiration. I need the component that is going to help me create the channel pump or the aquaporin or so on. So once you get that signal, your specific gene that codes for that protein is going to be activated, is going to be ready to get transcribed. And so you transcribe just that specific gene that your cell needs or your body needs, because sometimes it's going to be transferred in other places, okay? So a few things to remember. RNA is different from DNA, right? The first one is that doesn't have a T, it has a U. So where you have an A on your DNA, now your complementary strand is going to have a U and not a T, okay? So that is definitely a difference. And then we are going to see that the enzyme that does this addition of nucleotides, it's called RNA polymerase. So again, you know, not very different from the DNA polymerase. The DNA polymerase adds nucleotides during DNA replication, but here you're making RNA, so it's called RNA polymerase because it's doing pretty much the same thing, is adding nucleotide in the new strand. And very much so like the DNA polymerase, RNA polymerase also has to synthesize from 5 to 3. Okay, so the new strand is going to go from 5 to 3, exactly in the same way as the DNA polymerase was doing. So let's go in the details of each steps of transcription. We're going to look first on how transcription happens in prokaryotes, because as I was saying before, it's easier, it's less complex. So we're going to look first at the prokaryotes, and then we're just going to look at the differences in the eukaryotes. So what I don't cover in the eukaryotes means that it's identical in the prokaryotic process. Okay. Okay, so step 1, initiation, step 2, elongation, step 3, termination. Those are the 3 steps that we go through when we do transcription. So from DNA to RNA. Remember, this is what we're trying to do, is step 1 of the flow of the central dogma. So first thing to notice is this little thing here, promoter. So this guy here is where everything started, okay? Everything starts at this level. So let's go. One thing that I want to show you before we go in the details of initiation is that you see that now these strands are labeled. So this is called template DNA. This is called non-template DNA. It can also be called coding DNA strand. So it's the same, and I'll show you in a second. Okay, let's start with initiation. Remember, I show you that promoter, that little sequence on your DNA that was right at the beginning of your transcribe region. So it's before you start transcribing. And so the promoter is very important because it's a sequence on your DNA. See, on your DNA, these are your 2 strands of DNA that is going to be recognized by the RNA polymerase. And the RNA polymerase can bind to these specific sequences. It recognizes the sequences and can bind to the sequences, so then can start to transcribe the region that is in yellow here, which is your gene. So let's pretend this gene, this yellow area is the gene that codes for the color of my eye, okay? And we need more pigments, and so we need to transcribe this, okay? Okay, we are in prokaryotes, so it's not that gene, but just so you understand, the concept is the same, right? Okay, so we don't want to transcribe constantly genes if we don't need that protein. So the promoter is important because you can activate with a series of actions and feedback that can happen at the molecular level in the cell. You can activate or wake up that promoter. So now that promoter can send a signal, and the RNA polymerase can recognize the signal, can come, bind to the promoter. Once it does that, then it can start to, obviously, they're going to open the bubble like you saw with the replication. Is going to be similar here. Some enzymes are going to come and they're going to open the double strand, because remember, we're only transcribing one strand, right? mRNA has only one strand, so we're only transcribing this strand here, the one that goes from 3 to 5, because the RNA polymerase is going to add the new nucleotide in direction from 5 to 3, okay? So this is your promoter. If we want to look at the details of how this promoter looks like, these are the sequences. So usually this is how they are. There are 2 different sets of sequences. One is 10 nucleotides before the transcription start site, so your point 1, and the other one at 35 elements before. So this is your promoter here, and those are the ones that specifically bind with the RNA polymerase. This is in the prokaryotes, okay? We're going to see that in the eukaryotes, it looks a little bit different, but the concept is the same. After the RNA polymerase comes, starts, binds with your promoter, start transcription, you are going to actually start step 2, which is the juicy step, which is the elongation. So this is when you're actually making this strand. So you're going to have your RNA polymerase. Remember, this is in yellow. This is the part that we're coding, right? That we are transcribing. So it's our gene. See, it's small, doesn't have to be very long. It's only one portion. And so your RNA polymerase is going to transcribe, is going to add these nucleotides from 5 to 3. So it's already read this, and it keeps going. Very similar, the RNA polymerase is going to come, reads the nucleotide, grabs the nucleotides that is going to match, and it puts it down and binds it. And then it grabs another one, and it grabs it, and it grabs. So it reads and grabs nucleotides, and it puts them one after the other to make this new strand of RNA. Now, as I was mentioning before, the template of DNA that we use to create this new strand of RNA is called template strand. The other one, the one that is anti-parallel, the complementary one, can be called not template or coding strand. I like better coding strand because if you think about it, it makes sense. Because yes, you are creating this mRNA strand based on your template strand, right? So let's pretend this is the blue, right? Blue strand here, 3 to 5. So let's pretend this is here. So this is what is written, right? This is the nucleotides, the bases that you have in the template strand. This is what you're going to transcribe in your RNA, right? T-A-A-U, because remember that now you don't have a T because it's RNA. C-G-T-A-A-U-G-C. Remember that is, we talked about that when we talked about the macromolecule. So if you look at this and you look at the coding strand, you're going to see that the RNA and the coding strands are going to be identical, with the differences that when here is a T, here is going to be a U, because it's complementary to the template strand. So it's a genius way that nature found to, you're really coding, you're really, the code is really in here, right? This is what we are actually making, but because it can only go from 5 to 3, is using the complementary to make the RNA, okay? So it keeps going until it's going to see, is going to encode what is called a raw binding site. The raw factor is actually a protein that is going to give the signal to the polymerase and to the RNA polymerase that we're getting to the end of the transcription of that gene. We cannot transcribe the entire DNA, right? That would be, doesn't make any sense. We just want to transcribe that gene. So once you get close to the end, once it gets close to what we call the terminator site, there is the raw factor that is going to bind. So there is a raw binding site that is going to recognize the raw factor. It binds it and it gives a signal to say, we are done here. We are not going to keep transcribing. And that happens like this. So then you have your new mRNA and everybody is happy and you're done. Okay? This is in prokaryotes. Now, as I was mentioning before, in prokaryotes, sometimes you can have this process of transcription happening at the same time with translation. What does that mean? It means that while you're doing your elongation, right? So this is 5 prime, this is 3 prime. While you're doing your elongation, so this, you keep going. Here you still have your RNA polymerase attached to it. On that side that is done, your ribosomes are going to come and start translating that side. So it happens at the same time. And so you also can have multiple genes that are transcribed and translated at the same time in bacteria. So let's say, for example, that you are going to have also this gene here and this gene here. So this is, so the green here is your blue in here, so is your DNA, and your blue little woggly thing, those are your purple, so those are your RNA that are being transcribed, right? And they're coming out from your main DNA because they're still attached because you're still transcribing them. They're not yet in the terminator step. but even though they're still attached, the ribosome are going to attach to them, and they're going to start doing translation. So they're going to start reading that mRNA and creating the protein. So it can be, at the same time that you get transcription and translation happening in prokaryotes, so bacteria. Now, let's look at the differences of what happened in the transcription during in the eukaryotic cell. We already saw before some differences, nucleus and this processing. So the similarities is that exactly in the same way that we just saw with the bacteria, also eukaryotic cells have the same steps. So initiation, elongation, termination, no differences whatsoever in here. But we have some differences. The number one is the promoter. We're going to look at that in a second. We also don't have that fact that you can do multiple genes at the same time and doing translation as you do transcription at the same time. It's mono in eukaryote, meaning one at a time. And in separate location. And then our RNA polymerase is more complex. You actually don't have only one, you can have up to 3. And like we were saying, at the end of transcription, we don't get the mature mRNA right away like prokaryotes do. We actually get a pre-mRNA that needs to be processed. And this processing happens inside the nucleus, and we're going to see what these two processes are. So addition of the 5' cap and poly-A tail in the 3' end, and then RNA splicing. We're going to look at this in a second. So for now, these are just the differences. Everything else is the same between eukaryotes and prokaryotes. Let's look at the promoter first. So in eukaryotes, the promoter is actually way more complex. Sorry, we have what we call Tata box, roughly 35 elements before your transcription start site. So again, the yellow part is the gene that we want to transcribe. In this case, my analogy with the pigments of your eye, it's accurate because eukaryotes, humans are eukaryotes. So but you have to give that signal in the same way. We don't want to waste proteins. We don't want to waste energy. Our body, as always, is very, very sustainable and doesn't like to waste energy. So only if that promoter is activated, and we're not going to look in the detail of that, but only if that promoter is activated, it gives the signal to your RNA polymerase that now recognize the site is going to bind. You see that there are many different factors here. So it's very complex. It's going to activate itself, is going to bind, and then is going to start transcription. So transcribing just that yellow portion, which is the gene that we are interested in that moment. Your body wants that protein to be expressed for some reason. It needs that specific protein. So that gene, that yellow gene in this case, needs to be transcribed. Okay? So this is a picture from your book. It's the same concept. It's just it shows you in a different way how the promoter is different in eukaryotes. Okay? So then, I'm not going to go over again the steps of transcription because they're the same, so it would be, redundant. But we do want to look at this processing because we said, okay, at the end of transcription in eukaryotes, our RNA is not ready. It's called pre-mRNA. It's not mature yet. So what is going to happen is that we're going to protect our RNA, needs to leave the nucleus and go in the cytoplasm. So to protect the RNA from degradation, we're putting a 5' end cap and a poly-A tail 3' end on the 3' end. Okay, so it's just to make sure that we don't get degradation right away. If you ever going to work in the lab with RNA, you are going to see how fragile it is. it's actually very easy to degrade in the nature RNA because of its role, right? We don't really need this molecule to be stable and to stay for a long time. Remember, the analogy is just a piece of paper, is a post-it that you're bringing in the kitchen for the moment to do it, and then you throw it away. So your body cannot put a lot of energy to create this because it's not supposed to last. It's supposed to just, it's a messenger, right? This M stays for messenger. It's a messenger. It's bringing the message outside, in our case, outside of the nucleus, so we can do translation. But it's not meant to stay stable for a very long time. But we do need to be stable for to do the job. And so to make sure they do the job, they have this 5-cap and the poly-A tail to make it more stable. Now, the other thing that is interesting that probably you already noticed is like, why do we have different colors in the actual sequence, right? Obviously, in the DNA, you don't have, or RNA, they're not different colors. This is just a cartoon, but to kind of show you that some parts of this RNA, the one in purple, are called exon. Those are the ones that actually are coding for proteins, meaning they have codons, they have codes, and we're looking in a second what codons are. Those nucleotides are coding for the protein. The introns are areas that are not expressing anything. So there is still nucleotides in here, but they're not really expressing for anything. So it would be a waste to have them going through. So this is why we cut them out. Okay, so we cut the introns out, and then we splice the exons together. And so at the end, you have your mature RNA, and that is what leaves the nucleus and goes to do translation. Different pictures, same story. We put the 5-cap and the poly-A tail to protect our mRNA. Remember, this is still pre-mRNA. And then the one here that are depicted in white are those introns that we don't need. So we're going to cut them out, and then we're going to splice them together. And now you get your mature mRNA that is ready for the next step, which is translation. Okay? So this is a cute table, a nice table, maybe not cute, but nice, it's well done, where you can see the differences between bacteria and eukaryotes, so prokaryotes and eukaryotes. Some of this we already covered, others don't worry, but we didn't cover, don't worry too much. We didn't really go in the details of the core and all enzyme, that is fine. And just, you know, the promoter is different, obviously. Here is not mentioned, but it is. Okay, perfect. Okay, so here I post this awesome video. I love this video. It's only 2 minutes and 41 seconds, and it covers everything we are going to cover during this very long lecture. So it's very good as an overview, as that big picture that we always want to have clear in mind to watch it. So because it covers everything, see, the title is DNA to protein, so it's the entire central dogma. I encourage you to just stop me and go and watch the first part that covers the transcription, because it's going to give you, because I don't want you to start translation right away. I want you to understand first what we're talking about here. So go ahead, pause me, reread your notes about transcription, and watch at least the first part. You can even watch the entire thing, it's only 2 minutes and 41 seconds, and then you rewatch the translation again after we do the translation. But go ahead, pause me, look at your notes first to see if you are understanding what we're talking about, and then watch this short video, because it's really going to solidify some of the concept. Okay, I hope you did that you pause me, you watch the video, check your notes, and as always, remember, you don't have to watch the entire lecture all at once. You can pause it and take a note of which minutes you were done, and then come back. So some activities, this is awesome. I love this. It's a little table where you're going to put what is only belonging to transcription, what it belongs to only replication, and what is actually happening in both processes. Remember, replication, the goal of replication is different than transcriptions, but they do have some similarities, and so I want you to make sure that you understand and you put them in the right place. So for example, DNA polymerase, it catalyzes what? Right, replication. So this one would go here. You would write replication DNA polymerase. What about RNA polymerase? That only happens in transcription because you are making RNA. So you will put that there. Something that happens in both, they both have to go, the direction is both from 5 to 3. So you can put that here. Okay, so that is kind of the idea here, to do this exercise, and in that way you have it, you know where things go. Okay, so now we did DNA to RNA, right? We need to make the meal. Right? We transcribe, we got that recipe out of the bedroom. We are in the kitchen now with a Post-it, but we transcribe everything so we know that specific recipe, what we need to do to make that meal, that specific piece, not the entire, all the recipe of the planet, just that specific one. Now we need to do it. Now we need to make the protein. We need to do that phenotype, right? So I added here nucleotides and amino acid because it's important to keep that in mind. As we said, we're going from transcription and now we're going to do translation. Why I added this? Remember when we talked about macromolecules, we talked about building blocks, right? Each macromolecule has their own building block. RNA and DNA have nucleotides as building block. Protein has amino acid. So the scientists that discovered this process, or scientists, more than one that discovered this process, said, wow, we are going from nucleotides to amino acid. We are changing language, right? We are not going from DNA that has nucleotides to RNA that also has nucleotides. We're changing here. We are translating, okay? So from nucleotides, we go to amino acid, meaning we are changing language, meaning we are going to need a dictionary, right? If you learn a new language, you don't know what that word means in the other language. I need a code. I need a dictionary. I need something that tells me that word means this, right? It's going to be the same for translation. We're going to see that we have a dictionary, we have a code that tells us, okay, these 3 nucleotides are coding for this amino acid, okay? Because that is what we're doing here. We're translating. We have that code. We have that code went through transcription. We transcribe that, so we have it on our RNA. It's right there, but now we need to make the protein, so we're changing language. We need to do the next step. We need to see how we can go from nucleotide to amino acid. And we're going to see that we're going to have many different players that help in this process. And all 3 these players are actually made of different type of RNA. Okay, they're all 3 RNA based molecule. Okay, one is our friend messenger RNA. We just made it to transcription. We already know him, but we're going to see what are those other players. So, recap, I'm not going to spend any minutes in this one, but I do want to remember, right? We're going to talk about how we move from one language to the other. So from nucleotides to amino acid, make sure you remember how nucleotides are and the difference between if I'm RNA or if I'm DNA and so on. Same for amino acid. Remember what an amino acid is, which group it does have, and how we make proteins, right? We talked about this during the macromolecules. We said during the hydration synthesis, I'm making a molecule of water, H and OH goes away, molecule of water. Now nitrogen and carbon are free to do a new bond. This is called the peptide bond, but it's really a covalent bond, and so on, okay? And then we said amino acid. How many amino acid do we have? We have 20. They can be different depending on the special R side and so on, okay? Okay, let's get started. So now that we recap everything, let's see how it happens at the molecular level. How do we do that? How we do translation? Turns out that there is a code. Remember that I was like, we're translating from one language to the other. We need a dictionary. Well, we have it. Scientists discover, looking at translation and how organisms are making proteins, discover that there is a specific genetic code. Not just specific, this code is universal. What does that mean? It means that no matter who you are, if you are E. coli, a bacterium, or if you're a protist, or if you're an archaea, if you're a goat, if you're a dog, if you're a human, if you are a plant, no matter who you are, if you're a living organism, this is the code that your body is using to make proteins. So scientists realized after many studies that every time there was a certain sequence of 3 nucleotides in their RNA, that sequence was always linked to that specific amino acid. And so they made a table because it's easier, well organized, where they were able to check for every different type of sequence. We're going to see how we call these 3 letters together when it's like this. How is that code called? Every time they were seeing this sequence together of these 3 nucleotides, that was the amino acid linked to that. And this 3, that was the same amino acid linked to that, and so on. This is true for all 20 amino acids. So they discover, okay, with this 20 amino acid, which sequence are linked? What is that dictionary? What is that code? This is what it is. And it's universal, meaning that pretty much everybody use the same code to do that translation. Okay, so let's look a little bit at some terms here. So used by all organisms. So this is a set of rules that convert a nucleotide sequence to an amino acid. So that means, there is a code. All 3 letters, 3 nucleotides equal 1 specific amino acid. And as we said, so that what that means is that flow of information that we said, DNA, RNA, protein, is really based on a triplet code. So 3 nucleotides code for 1 amino acid. Where is it? Here. 3 nucleotides code for 1 amino acid. That triplets, those 3 nucleotides, is called codon. Okay? So how do we read this table? Because if I give you a table like this, you need to be able to read it. Let's say that I'm going to tell you, okay, if this is your mRNA, and this is your sequence of mRNA, which type of amino acid you're going to get? You can do it because it's easy. It's like a game, right? So let's say the first letter, you would look always at the codons. So you would look for the first letter is here, and we're going to do an exercise in a second, so I'll show you better in there, but this is where the first letter is. So every codon that starts with a U will go here. Every codon that starts with a C goes here, with the A here, with the G here. Every codon that has a U as a second letter is under here. That has a C as a second letter goes here, and so on. And then for the third letter, you will see, okay, if my third letter is a U, I'm on this line. If I have a C, I'm on this line, and so on. And the same is for all of that, okay? So some things that we need to pay attention to this is that you probably already saw that we have only 20 amino acids, but we have 64 triplets, right? What you can notice here is that some codons, even though they are different, they still code for the same amino acid. So there is some type of redundancy in the genetic code. Do you think this is a good thing or a bad thing? Think about it. It's a good thing, right? And the reason why it's a good thing is, remember those mutations that we were talking about last time, those silent mutations, the one that they can change the last nucleotide and maybe we miss that for some reason. Even if that happens, it doesn't matter because they're still coding for the same amino acid. So this is a way to protect us, right? It's a way, in a way that where the third position, it's called wobble position, meaning that sometimes that nucleotide can be different, and that is fine because it's not going to affect your proteins. It's not going to affect your amino acid. It's not going to affect your phenotype. So that is really good, okay? So that's the first thing that I want you to pay attention to, the fact that we have 64 triplets, 20 amino acids, so meaning that some triplets code for the same amino acid, and that is good, right? It's not going to affect our amino acid. The other thing that I want to point out is not every triplet is coding for an amino acid. We actually discover that there are 3 codons, right? 3 triplets that don't code for anything. They code for a stop factor. So that means that if you get that in the sequence of your RNA, you get the signal that says, we're done. No more amino acid needs to be added to this protein. We're all done, okay? That is what that stop codons means. And then there is this in yellow, in red, that is the AUG codon. That codes for methionine, always and only for methionine, and that is what we call the start codon. So every time you have an AUG, that is when your translation is going to start. It's the signal of this is number 1. This is your start codon, okay? Let's do this together. I want you to practice. So obviously this is going to be your mRNA in black here, and this is the nucleotides that we transcribed, right, during transcription. Remember, we just did transcription. We went through processing and everything. This is your mature mRNA. And now we need to see, based on what we discover, knowing that this is our universal code that we're going to use, which type of amino acid we would get in our polypeptide, in our protein, based on this. So remember how to read the table. First of all, remember that we read 3 nucleotides at the time. That is your codon. That is what codes for a specific amino acid. So the first thing is to make sure you say, okay, these 3 go together, these 3 go together, these 3, this 3, and this 3. Once you have that clear, now we need to find the nucleotides in the table. It can be overwhelming, but if you follow these directions, it's always going to be very easy. Because remember, when scientists discovered the table, they did it in a way that we could use it easily, right? Where we can say, oh, okay, I have that nucleotide on this mRNA, which amino acid I'm going to get? Easy. You always start from the first letter. So the first letter is an A. So you go down here, it says, oh, first letter, oh, it's an A. It must be in one of these 4. Perfect. So we already know we need to, it's like looking at a map. We already know we need to be here. The second letter is a U. So you go second letter, U. Perfect. So first letter A, second letter U, you know you're right here. This is the square you need to look at. And then you look at the third letter. Oh, that is a G. So you go third letter, you know you're here. Third letter, G, A-U-G, methionine. Okay? So then you would write down here, methionine. Okay? So I give you a couple of minutes to do this on your own, and then we can look at the results together. But take a second, do the same for ACA, AAU, and so on, okay? Start with the first letter, then the second, and then the third. Okay, I hope you paused the video, I hope you did it, and then let's see at the solution. So this would be the amino acid that you should have in your notebook. Notice that the UGA is the stop amino acid, meaning we're done. That is our protein, okay? So we did translation. Let's go ahead and do the second one. You guys can do it on your own. Remember, we always do that first. So those are our codons. And then this would be your solution. So now our UGA is up here. So that means we are now going to keep translating this because our codons stopped here. There is a stop release factor. We're done. Okay. This is great, right? This is so easy. Does it happen like this in real life in our cell? I mean, the translation part, the code, yes, but the molecular level is way more complex, right? Like how our mRNA actually read to make the protein, right? How do they do it? Like how does that machinery work? Because there is no table in our cells. There is no little human that goes and says, oh, this goes here, this goes here. Of course, things are more complex. That is how the code works, of course, yes, but how does actually happen? How does translation actually happen at the molecular level inside our cell? Let's look at it. So first of all, as I told you before, we have 3 major players. These players are all RNA based. And we call this the protein synthesis machinery. Protein synthesis is the same thing that what we're looking at. The protein synthesis is this process of like doing transcription and translation is really the synthesis, is the last part of synthesis of protein, making proteins. So we have mRNA, ribosome, and tRNA. Remember, we have differences depending if you're prokaryotes or eukaryotes. That matters. So we are in the cytoplasm in both cases at this point. We are down here if you're a eukaryote. But remember that prokaryotes can have that happening at the same time while transcription is happening. So just remember that there are some differences, obviously. So let's start with the mRNA. So mRNA, we made it through transcription, right? So just saw it that in a second ago. Inside the nucleus if you're a eukaryotic, outside the nucleus if you're a prokaryote. And this guy here is our mRNA. So it's the orange strand. So the mRNA is the one that actually carried the instruction for making proteins because you just transcribed them, right? Remember, you just took the book with all your recipe, put it on a piece of paper, that is your RNA, and then you moved it. So it's your messenger RNA. This M is actually for messenger because it is bringing the message, is bringing the code, okay? It's the one that actually has the instructions to make your proteins. And so he has the codons on it. So remember those codons that we just saw, the 3 nucleotides, 3 nucleotides, 3 nucleotides. This is where the codons are. They are on your messenger RNA. Okay, and so right here, we are going to see that then we have also our ribosomes, this big blob in gray, and then we have tRNA that is in purple here, okay? And they all work together to make our polypeptide here, which is your protein. Remember those peptide bonds that we just said, and these are amino acid and tRNA. Okay. So let's start with the ribosome. The ribosomes, a little bit of recap. Remember, when we looked at the prokaryotes versus eukaryotes, we talked about ribosomes briefly. We said they both have it, right? They're here, ribosome, and ribosomes in the eukaryotes are often associated with the endoplasmatic reticulum RAF. So RAF because has those ribosomes attached to it. So they both have it. So a ribosome is a big structure, is a big complex molecule that is responsible to really do that translation. So really the ribosome is the structure that goes and reads the mRNA codes and say, oh, I need that protein. I need that amino acid. This is the codon I'm giving the signal, and it's really the where the magic happens. So it's a complex molecule that is made of RNA. It's actually called rRNA because it's ribosomal RNA. And also have proteins connect to it, but the proteins are just structural here. So what really does the job is the RNA portion of the ribosome. And so it has 3 sites. It's called A site, P site, and E site. The E site is the exit. The A site is where your tRNA comes at the beginning, and here is where your chain starts going, okay? Has 2 subunits. One is a large subunit and the other one is a small subunit. Prokaryotes and eukaryotes have the same structure, meaning that they have, both of them have ribosomes that have a large and a small subunit, but the dimension and how the type of RNA that they have, rRNA that they have, is different. And that is important because when you do molecular work and you want to target, for example, a prokaryote and not a eukaryote, you can target your genes that are coding for this RNA, and so you can target different prokaryotes. You know, you can distinguish prokaryotes from eukaryotes, but also if you want to inhibit, for example, translation of some proteins that a bacteria is doing, you can target the ribosome, the bacterial ribosome, and is not going to impact our ribosomes. So it's going to impact and damaging these bacteria that maybe are making us sick without damaging our ribosomes. So we can still do translation, but the bacteria can't. So that is really good. Okay. So we can use, as always, we don't invent anything in biology. We apply our knowledge. We discover things, and these things that we discover with the knowledge that we are gaining, we can use this knowledge in our advantage. Okay, so another thing that is super important, each of these sites has a spot for 3 codons. So 3 codons are going to be here, 3 codons are going to be here, 3 codons are going to be here, because remember your messenger RNA is going to be here like kind of in a sandwich right there, hugged by the 2 subunits. And so the ribosome is going to read and move 3 codons at the time from 5 to 3' end, as always everything does, okay? So that was the ribosome. The third player, which is my favorite, is the tRNA. The tRNA is called T for transfer, so transfer RNA, but I like to think that T, even though that is not what that stands for, is for translating, because really the tRNA is the adapter, is what is doing the translation. Because how does it work? So first of all, the tRNA has a high specificity, very high. So you have between 40 and 60 different tRNA molecules in your cytoplasm that are there hanging out right there in the cytoplasm. And then you have your amino acid in the cytoplasm, right? They're there. Everybody is having the time of their life hanging there. When there is a signal that comes from the ribosome that says, hey, I need this specific amino acid because I just read the codons on the mRNA, and that codons is coding for that specific amino acid. It's going to send the signal to the aminoacetyl tRNA synthetase, which is an enzyme, and this enzyme is going to grab that specific tRNA and that specific cysteine, amino acid, in this case the cysteine for this anticodon, and is going to put them together using ATP, and we call this charging the tRNA. So this happens in the cytoplasm, far away from your protein synthesis machinery. In your cytoplasm, there are going to be feedback and some type of signals that once the ribosomes reads a specific codon, gives the signal to say, I need that amino acid. So this happens where the tRNA that has an anticodon, so that means that also is the same codon that matches that specific amino acid, right? Is anticodon compared to the messenger codons. And I know this is a lot. We're going to look at that in specific in a second. Let's just say for a second here that those two match. They always go together. And so during this process that you need ATP for it, you're charging, you're putting these two together, the tRNA and amino acid. Okay, so they become a complex. You can read this here is what I just said. So once the tRNA is charged, meaning it has the amino acid attached to that part, the 3' end, then is going to go inside, pretend that here you have the ribosome, is going to go inside the ribosome in the specific site and is going to bind with the mRNA, okay? That is what I was saying a second ago. You have those codons in the mRNA. Remember those codes that code for a specific amino acid? These codons are going to recognize the anticodon. As always, they're going to be complementary. And that anticodon is the one that is carrying that specific amino acid. So at the end, that specific amino acid is linked with this codon, but also linked with this anticodon, because remember, everything is complementary and antiparallel. So it's a genius way that nature found to make sure you can still go from 5 to 3 end, moving in the right direction, connecting by using like an adapter molecule that can come with the right amino acid and connected with that codon here. Okay. Perfect. And yes, the tRNA is binding with the ribosome, and we'll see that in a second. So let's see actually how this works. So translation has three different steps, very much like transcription. You have initiation, elongation, and termination. During initiation, your complex machinery is formed. So you're going to have your first tRNA charge with amino acid. Remember that this is the methionine, right? This is your start codon, AUG. So your anticodon in the tRNA is going to be U, A, and C, and it's going to have a methionine attached to here. So this is happening. Remember, we just saw when you charge with the enzyme, ATP is used. You're charging this. You're going to have your small subunit down here, this large subunit of ribosome subunit up here that is going to hug your RNA and is going to read in this direction. Okay, so the complex, initiation complex is completed, and this tRNA is going to go inside your RNA, ribosome. Once that starts, then you're going to start to have elongation. So remember, each site, so this is A, this is P, this is E, reads 3 nucleotides at the time. So those codons on the on your messenger RNA. So it's going to start and it's going to then say, okay, then it's going to start reading and says, okay, next one is this. I don't know, UUA. I'm making it up. So it's going to give a signal and it's going to call another tRNA with that specific amino acid that's going to come, go in the A site. The chain that has been formed in the P site is going to be moved in this one. Because this moves, now the tRNA doesn't move, but the ribosome moves, and so what was in A goes in P, what goes in P goes in E. So in that, it's a way to get rid of the tRNA without the chain. So the chain moves to this one, and so this one with all of it is going to move in the next one. Okay. So elongation is a way for the machine, the ribosomes, to move and read one codon at the time, the messenger RNA. So your ribosome is reading the messenger RNA one codon at a time from 5 to 3, and is going to say, hey, this matching tRNA needs to come and bind to that codon that is exposed. And it keeps going, and the existing amino acid chain, which is this one, is going to go to the new amino acid on the new tRNA, which is this one here, and then keeps going. Okay? So this is a different way to see this. I like this because it puts the letters of the sites, so it you know, you can kind of see the movement. So it's the same thing. The new one comes to A, is going to bind, so the old chain that is being formed is going to bind and go to the new tRNA, and then now it's here. Now this moves from 5 to 3, so it's going to push the tRNA. This one is going to go to the exit and go out, and the P is going, and this one goes on the P, and now you have a new spot available, and that goes there. Okay, so it keeps going, keeps doing this until you reach the stop codon UAG, and now you get no tRNA, you get a release factor. It says we are all done, there is no new amino acid. So when that comes, you actually get a signal where the polypeptide now get released, and everything is then recycled, and we can restart again in case we need to, maybe for something else. Okay, and your protein is all done. Remember, each dot is one amino acid, and so this line is the peptide bond, the course between each amino acid, and now you have your protein all done. So this is just another picture of this termination part where, you know, you have the release factor and then hydrolysis happens. So remember this, we all looked at this, hydrolysis, so the components are dissociate and we can reuse everything. And we use a GTP to do this. Okay. So this is a little activity I did. It's not perfect. I made this. I apologize if it's confusing. I hope it's going to make it clearer. So this black one here is from 5 to 3 and is your messenger RNA, the mRNA that we just transcribed, okay? These guys here are your stop codons, okay? Are your, are your tRNA that are all over around the cytoplasm. This guy in gray is your ribosome, okay? So it's hugging the tRNA, the mRNA. Okay, the first one that comes is the methionine because the IUG. So the ribosomes reads and says, we need to start translation. So it calls that tRNA that is already charged in the cytoplasm, and it tells it to come and to bind to the AUG, okay? And then it reads the next one, it goes to the next level, and is going to say, okay, next free codon is this one, CCU. So I need a tRNA that has an anticodon that is GGA, which means it's going to have the pro amino acid attached to it. The next step is that bonding, right? The covalent bonding between the two amino acids. At this point, the ribosome moves because it needs to keep reading. And so it goes from 5 to 3, moves, but remember, it moves always 3 nucleotide at the time. So now you have this original tRNA on your exit, and this one here that is bonding, right, on the P. So this guy goes away. Okay, the tRNA is in the exit, is going to go away, but the methionine stay attached to the new tRNA because you're starting creating this polypeptide. Okay, so now the ribosome, it says next is GCA codon, which means I need a CGU anticodon. So it calls that tRNA that has that anticodon on it and has that specific amino acid, because remember, that is genetic code. This codon is coding for that specific amino acid, okay? So same story here, we're going to create that bond. So now you have 3 amino acids that are linked between each other through peptide bond, and the ribosome is ready to move again. Remember, 3 codons at the time. So again, now you have that chain linked to this guy here on the P site, and the old tRNA is on the E site. So that goes in the exit, is not going to stay. So now we are ready to get a new tRNA. The same story is here. The codon on the 5, remember it's 5 to 3, is AAG. So we need a UUC anticodon on your tRNA that was charged in the cytoplasm. We call it, it comes, creates the bond, and moves again. Same story here. Okay? Moves again. So now this guy is in the axis side, so it gets rid of, we just get rid of it. And now what happens? UAG is the next codon that ribosomes is going to read. But that is a stop codon. So at this point, we're telling our machinery, we are done. There are no other amino acid linked to this one. We have a stop codon here. So it's time to get rid of everything. So your polypeptide is ready, it's going to be released, and everything is going to be reused. Okay? Once your protein is ready, so once your protein is all done and released, you are going to see, is going to signal peptide is removed and your protein is going to be released. Remember that we are going to go through protein folding, modification, and targeting. I'm not going to cover this part again because we did talk about this when we talked about the different components of eukaryotic cell. Remember, we talked about the ER, RAF-ER, the Golgi. So that is when this happens. After you do, and remember, we talked about how protein get modified. So this is just your linear protein, polypeptide, but can become secondary structure, tertiary structure, sometimes quaternary structure, depending on which protein you're making. So that happens, all that happens after after the translation is done, okay? And so at this point, this is when you can do some connection with other things that we cover when we talked about, okay, so what happens, to my proteins. Now it needs to do the job that needs to do. So if I made an enzyme, the enzyme needs to be moved and needs to be brought where it needs to go to do the job. right? If I'm making a component of a channel, I need to bring this component out, and remember that how we bring in and out components that are too big. We use exocytosis, right? So put all your information together, but that is after you make your proteins. So I love this because it's a little table where it kind of goes through the differences between translation in prokaryotes and eukaryotes. As you can tell, they're very similar. The difference is the ribosomes have a different structure. So this is the weight of those subunits. So it's different in bacteria and eukaryotes. Also, the actual genes are a little bit different. They're going to tell you, how the initiator are, the fact that we don't have in eukaryotes simultaneous transcription and translation, which we already talked about that, and this is the picture of that right here. This is prokaryotes. In prokaryotes, you do have that happening at the same time. We don't have that in eukaryotes. Okay, again, the same video as I told you before, super well done, very cute and fat. I shouldn't say cute, but very clear and short. Very good overview. I encourage you to watch it again if you watch it all before, or watch just the translation part. And then we have some activities. So like you did before, here now you have all three processes: replication, transcription, and translation. Remember, replication happens during interphase. Transcription and translation are part of the protein synthesis gene expressions. So it's where you want to transcribe, you have one specific gene that needs to be active, that is activated, because now we need that gene to express that protein, to synthesize that protein. Okay, so original instructions, I want you to remember that they're different. So replication is nucleotides, and there are 2 strands of DNA that are going to be replicated, right? Transcription is still nucleotides, but is only 1 strand. Translation is the original instructions is written in what? Still a nucleotide, right? Those codons. But the final product here is amino acid, while in this case is still nucleotides. And then the location. And then this is filling the blank, is super short, like very synthetic, but it kind of, big picture. So you think about something cannot leave the something. Right, the DNA cannot leave the nucleus. Very good. And then you can try to do the other ones together by yourself. And then the last one is the finished product is a protein. Very good. So now that we know everything about everything, so we know how we make our proteins, I want to go back 1 second and talk about mutations. And the reason why is because often we give a very bad connotation to mutation, because mutation can be harmful. 100% yes. We talked about this. If you have mutation, you can have cancer, you can have genetic disorders, you can have, many different things that are definitely harmful to the body and to the organism, 100%. But also, we said at the beginning of the class, and we talked about this also when we talked about genetics and allele and variation, that mutations are also the source of the rich diversity of our genes in all living organisms. They're not always harmful. First of all, some mutation has no effect whatsoever to the sequence, because remember we said some silent mutation, the redundancy of the genetic code. This doesn't matter that it's the wobble position. Even if I have an A or a T, it doesn't matter. I am coding for the same amino acid. But sometimes I am changing the phenotype, but not because it's a giving harm to the organism, it's giving variation to the organism. We're all different. I have green eyes, you might have brown eyes. I have white skin, you might have brown skin or black skin, or I'm tall or short, or I have my ear lobe in a certain way. Those are all mutations. Those are all alleles, right? The variability of those alleles. That is why how beautiful are the diversity in our world, in our planet. And this is just talking about humans. Think about everything else, all the living organisms, all the different species, and so on. Is how evolution can happen, is because you have mutations. If you don't have change, which is a mutation, you would not have evolution. We would still be all that original bacterium, right? You have no change. There is no mutation. There is no evolution because there is no natural selection, okay? So mutation contribute to the process of evolution by natural selection. Of course, they can be harmful 100%, but they're also the rich source of diversity, okay? And this is what makes our planet beautiful, because everything is different. So I post this video here. I love this video. And I'm going to put also a cross here on the note, because you do know all of it. By now, everything that is shown in this 5 minutes video about DNA, we cover it all during this entire semester. So go ahead and watch it, and you're going to feel so proud of yourself because you know all of it. And it's just a cute little video that gives a good overview about pretty much everything we cover at the molecular level. And with this, I want to tell you, congratulations, you made it. We finished the semester. This is the last lecture. We went through everything that we could have in this amount of time about the molecular level of our cells. So we said at the beginning, we're going to cover atoms, molecules, and cells. And that is what we focus on. We really went to the details of how our cell works at the molecular level. And so I just want to say that I'm happy that you guys did this journey together with me, and I hope that now that puzzle that we started at the beginning is clear, that you have the clear picture of all the little pieces that we put together, how you can see how everything is connected. That of course is complex. Science, biology is very complex, and we try to simplify some of this concept, but I hope that you can see now how these organelles work together, how things work together, how processes work together. And I hope you learn not only the content, but also important skills that are going to help you to the next, for your next step, because our journey together is over, but your journey just started. This is just the beginning for you. For most all of you, you're going to go ahead and keep studying and keep following that why you're doing this. And I really wish you, the best luck, and keep in touch and tell me how things are going. Okay, guys, it was a pleasure to be your instructor this semester. I hope you had fun and you learned a lot, and I'll see you around. Bye, guys.